Hello everyone, my name is Vedant Karalkar, and my project focuses on using a biosensor to monitor environmental stress in space-grown plants. So humans are innate explorers, and our next mission takes us further than we've ever gone before, to Mars. The key in space exploration lies in plants. Plants are crucial for astronauts' survival aboard the ISS. Plants help astronauts by recycling their air, water, and waste materials, as well as supporting crew diets on longer missions. And as we plan to travel further to Mars and beyond, we will only be relying more on plants for our survival. For this reason, monitoring plant health is essential on the ISS, as plant death can endanger the lives of our astronauts. However, in some cases, plant health can be extremely unpredictable, and this can be seen through the Zinnia hybrida plants. After just a couple weeks of growth on the veggie chamber aboard the ISS, the plant showed severe necrosis and complete plant death. Zinnia plants are good model plants for environmental stimuli, meaning that other plants will respond similarly to environmental stresses. Such unexpected plant death could be fatal for a crew if the crew were to be relying on that plant as their primary source for food on a long-term space mission. Later analysis in literature showed that this was due to a flooding stress that the plants faced and showed that the observed leaf curling and root hypoxia was linked to a buildup of ethylene in the plants. So, you may be asking, what is ethylene? Ethylene is a phytohormone that plants produce to respond to various biotic and abiotic stresses that they face. However, high levels of ethylene can be hazardous for both astronaut and plant health. For this reason, ethylene is scrubbed on the ISS, making it extremely difficult to be detected. Thus, a fast and reliable monitoring of ethylene on the ISS must be created. Currently on Earth, we use gas chromatography and photoacoustic spectroscopy for the monitoring of ethylene. However, this requires for big, bulky, and expensive materials that are not sustainable for spaceflight. For this reason, we currently freeze plant samples and send them to Earth for further analysis. However, this fixation process causes for alterations in the biology of plants through nonspecific stress responses. These are limitations that will just be magnified as we plan to travel further to Mars and beyond. For this reason, in my project, I propose a solution, using biobits for the on-site fluorescent monitoring of genes in plant samples. This will revolutionize the way that we conduct spaceflight analyses by allowing for on-site plant health monitoring aboard the ISS. This is essential as we plan to travel further and will be relying more on plants for our spaceflight. As I mentioned, plants produce ethylene in response to various environmental stresses they face, and ethylene causes for gene expression changes that can then be monitored. Under the flooding stress, ethylene causes for the transcription of the GCN2 gene. This allows for adaptations in the plant that allow for better survivability, and thus the GCN2 mRNA can be used as a biomarker for the plant's survivability under flooding stress and the ethylene production under this stress. Under other environmental stresses, ethylene follows a signaling pathway, resulting in the transcription of the EBF2 gene. This way, the EBF2 mRNA can also be used as a biomarker for the production of ethylene under environmental stresses. This brings me to my hypothesis, which is that environmental stress in space-grown plants can be measured by looking at the changes in transcription level for EBF2 and GCN2 mRNA. In my project, I use a direct mRNA extraction technique instead of the current technique for mRNA extraction from plant samples, which is grinding. Grinding is extremely difficult on the ISS, and thus in my project, I propose the usage of a gold-plated needle that is coated with the captured DNA. This needle is so thin that it can directly penetrate through the plant's cell wall and obtain the genetic material, which will be captured using our captured DNA. This target mRNA can then be further analyzed, and this method will allow us to revolutionize the way that we conduct spaceflight analyses by allowing for the on-site mRNA sampling and thus the on-site plant health monitoring. This is also very useful for future spaceflight. This brings me to the overview of my experimentation. In my freeze and fly experiments, I'll be looking at environmental stresses through the EBF2 gene. I'll be doing this through a two-part fluorescence experiment, using qPCR for quantitative analysis and quantifying the stress that the plants are under, and biobits as a point-of-care sensor for detecting the production of ethylene under this environmental stress. Now for my ambitious experiments, I'll be looking at the specific stress of flooding, and I'll be using the same two-part fluorescence experiment, using qPCR for quantitative analysis and biobits as a point-of-care sensor. In my freeze and fly experiments, I'll be using Arabidopsis wild-type Columbia seeds. I'll have two groups, a control group that will be grown in a regular growth medium and an experimental group that will be grown in a medium with added ACC. This ACC will induce ethylene production and thus represents environmental stresses a plant faces aboard microgravity. Now, after 9 days of growth into the seedling stage, the mRNA will be extracted, and this will be placed into a PCR tube containing RT-qPCR reagents. This will then be used by mini-PCR to produce cDNA, which can then be frozen and sent to the ISS for further analysis. On the ISS, quantification of the cDNA can happen in 2 ways, the first way being qPCR. For qPCR, cybergreen binding dye will be added for the fluorescence analysis of the amount of EBF2 cDNA present, and thus the ethylene production under the environmental stress. Now, the second way is through BioBit's toehold system, which I'll be explaining more in my ambitious experiment, but allows for the fluorescent detection of the EBF2 cDNA and thus the ethylene production under environmental stresses. I anticipate to see the following results from my freeze and fly experiment. For my experimental group, I plan to see fluorescence for the EBF2 mRNA over time. This is because the addition of ACC in the growth medium will induce the production of ethylene by the plants, which will cause the ethylene signaling pathway to be activated and thus for the transcription of the EBF2 mRNA. Now, for my control group, I don't plan to see any fluorescence because I don't plan to see any production of ethylene. For my ambitious experiments, I'll be using the same seeds, but I'll be looking at flooding stress and thus the GCN2 gene. After sending the dormant seeds to the ISS, they will be grown for 9 days in a growth chamber, after which they will be split into 2 groups, an experimental group that will be subject to flooding stress for 40 hours, and a control group that will not. Additionally, I'll be running an earth control here at the ISS Environmental Simulation Lab. Now, data will be collected in the following method. First, the target mRNA will be extracted, and this will be placed into the mini-PCR system to produce GCN2 cDNA. This cDNA can be quantified in 2 ways, and the first way I'll be explaining is the BioBit's toehold system that uses BioBit's translation. First, the cDNA will be put through BioBit's cell-free transcription to produce GCN2 mRNA. This mRNA will then be subject to the toehold system. A toehold system is a pinched RNA system that allows for the fluorescence on-off detection of a target mRNA. In the absence of a target mRNA, the toehold system will remain in its pinched form, and thus a ribosome will not be able to translate the reporter gene. However, in the presence of a target mRNA, such as GCN2 mRNA, the toehold system will unpinch, allowing for the ribosome binding site and the start codon to be exposed. Thus, a ribosome will be able to translate the reporter gene, resulting in the production of a fluorescent response. This means that by simply adding GCN2 cDNA to a lyophilized BioBits and toehold system, we can fluorescently detect for the presence of GCN2 cDNA and thus ethylene production under flooding stress. Additionally, I'll be using qPCR using the same cybergreen binding dye for the fluorescence and quantitative analysis of the ethylene production under the flooding stress. I anticipate to see the following results. For my experimental group, I plan to see fluorescence for the GCN2 mRNA over time. This is because the flooding stress will cause for the production of ethylene, which will then transcriptionally activate for the GCN2 gene. In my control group, I don't plan to see the production of ethylene due to the lack of stress, and thus I don't plan to see any fluorescence. This can also be correlated to the survival of the plant under this flooding stress, making this biomarker so versatile and useful. This brings me to my conclusion, in which my project has detailed the detection of ethylene using a user-friendly, rapid, and on-site method. This will revolutionize the way that we conduct space plant analyses and plant analysis here on Earth by allowing for analysis to occur on the ISS and health monitoring to occur on the ISS. This is the first type of project to incorporate the RNA isolation method that I've detailed in combination with mini-PCR and the BioBit system, and this will revolutionize the way that we conduct plant analyses by allowing for on-site mRNA extraction and thus on-site plant health monitoring. Allowing astronauts to detect plant stresses at an earlier stage will allow them to perform quicker action, which can possibly save the lives of the crops, as seen in the Zinnia hybrida case. In the future, I want to look at the usage of a panel test for various other phytohormones, allowing for a more holistic plant diagnosis and a better understanding of plant health. Additionally, I want to look at the usage of an RPA system in combination with my BioBits system to allow for the isothermal and more efficient usage of the BioBits toehold system. And this is very important because the methodology that I propose in my research can be used here on Earth to be able to detect plant health and monitor plant health here on Earth using a very low resource and low-cost method, and this could replace the much more expensive GCMS methods that we currently use for plant analysis. I'd like to thank my mentor, Stuart Adamson, as well as my advisor, Dr. Natasha Haveman, for guiding me through this project, as well as the Genes and Space team for providing me with this opportunity. And I also like to dedicate this project to my grandmother, who inspired me to pursue space science and inspired me to compete in this competition. Thank you.